All right, so yeah, as Naresh said, my name is Sarah, and I'm really excited we'll actually be able to do this kind of like a presentation in person again, so that's pretty cool. And yeah, I'm excited to share the results from the research that I've been doing over the past 2 and now going into 3 years, which has started to look at the effect of different biofertilizers, which I'll define just as non-synthetic, non-chemical fertilizer options that can include organic matter, nutrients, and or plant beneficial microbes as well, on biomass crops to try to pursue a environmentally sustainable development of the biomass crop industry in Ontario and then throughout Canada. So just a little introduction to who I am. My background is in ecology, which is the study of the interactions between living and non-living components of the environment. And then during my undergrad, I took a course about agroforestry taught by our lovely moderator, Dr. Naresh Devathasan. And in that course, I was inspired to pursue a master's research project with him. And the project that we decided on was an expansion of his previous student's work, Sean Simpson. So Sean had done some preliminary field trials and greenhouse experiments with biofertilizers. And then we took those field trials and expanded it to larger plots and more fields and more different biofertilizer products to try to determine how different commercially available biofertilizers can influence the plant growth and yield, soils, and also the climate impact in biofertilizer or biomass crop production in Ontario, with the ultimate goal being to try to determine if some of these might be viable for use in commercial biomass operations. So just starting to get into what those trials actually looked like. So we included 2 herbaceous biomass crop species in our trials, which I'm sure we're all quite familiar with here. So we have switchgrass, which, as Naresh mentioned, we had the cave and rock variety, since that's what we had as mature plantings available. And then we also have miscanthus, specifically miscanthus saccharophloris. And both of these species are perennial, high-yielding C4 warm season grasses, with switchgrass being native to North American prairies, and you can see its range there in the map. And then miscanthus was actually introduced from Asia, and you can see where it's been introduced to in North America, also on the other side. So next we included 3 different field sites in our trials, and our first one here is the Guelph switchgrass field site, and I hope it's showing up well enough in the back. Our 4 plots are kind of outlined lightly in white. Those are the ones we actually ended up using for the trials, and this is a University of Guelph owned research facility just about northeast of the university campus. Next we have our Burlington switchgrass field site, which I don't actually talk too much about today, just due to the fact that we were only able to use it in 2019 because we had to reduce our research capacity in 2020 just to comply with the COVID restrictions on research activities. But this one's a really cool site because it is a grower-owned property by an OBPC member. So that was really great to be able to include as well. And then lastly, we have another grower property, which might look a little bit familiar, which is our Burlington miscanthus field site. It's here. Yeah, we were able to use this little strip of miscanthus, which I think we'll be looking at later today, in both 2019 and 2020. So we're really grateful, especially to be able to have these grower-owned properties, because that's pretty unique to these kinds of trials. Getting into the treatments, so we had 5 fertilizer treatments that we used in each of our study years, which are 2019 and 2020. So first we've got our zero input control, which we included at all sites in both study years, and we just allowed those plants to grow naturally, and also a chemical nitrogen fertilizer treatment, which was 60 kilograms of nitrogen per hectare of just granular urea. And those 2 treatments kind of counted as our basis for comparison for our biofertilizer treatments, because it's somewhat of the status quo in terms of how biomass crops are being managed at the moment. Next we've got Jumpstart. So moving to the other side of the dotted line, it's where I've kind of organized our biofertilizers. So in 2019, we applied Jumpstart, which contains a live fungus called Penicillium belli, which basically works to solubilize or to loosen up phosphorus that's gotten tied up onto soil particles and make it easier for plants to uptake. And with that treatment in 2019, we also applied a half rate of urea fertilizer to prevent a severe nitrogen deficiency, which could be possible from interfering with our ability to see the effects that Jumpstart actually has on the plants. Next we have MicPro, which is another, it's an inoculant. So we're applying another kind of a live fungus into the field, and this one contains Glomus centroidices, which is a fungus that lives in and around plant roots and essentially extends the root system to make it easier for plants to access more nutrients and then in more areas of the soil. So that was a granular product that we surface applied, also with that half rate of urea fertilizer to prevent a potential nitrogen deficiency from masking the effects of the MicPro product. And finally, we have Listagrow, which is a high solids liquid organic biofertilizer that's derived from municipal sewage. So we ended up applying that by hand at about 60 kilograms of nitrogen per hectare, just because the size of our plots was pretty small, but normally that product would be injected below the soil surface to control for some of the odor associated with it, and also to prevent from the nutrients being offloaded as gases, then you're losing half of what you put onto your soil. So that was our 2019 treatments there. In 2020, we did have to switch up some of them just due to some of the results that we got in 2019, and also to comply better with the COVID restrictions. So that zero input control and 60 kilograms of chemical nitrogen as urea kept the exact same from both years. Jumpstart is also relatively the same between 2019 and 2020, except for that we tripled the application rate and then also removed that half rate of urea, just to further emphasize the effects of the fungus itself, that phosphorus loosening fungus. Then below that, we can see that MicPro has switched over to Agtiv, but that's actually pretty much the exact same product. It's produced by the same company and contains that same kind of root extending fungus, with the only difference being that Agtiv is the agricultural grade version of the product, so it's the one you can buy more effectively, I guess, in bulk, and it's a higher quality version of the same exact thing. And we applied it at the same rate as well in 2020, so the only difference is really the name, and then we also took off that half rate of urea. And then lastly, I guess our most significant change from 2019 to 2020 is that we replaced Listagrow with two products by Vischer Holland, which is that little VH logo on the bottom, called Optimike and More, which are products that contain a mixture of different plant beneficial fungi and bacteria. And the reason we had to do this is just because of the manpower kind of required to apply the Listagrow wouldn't allow us to really distance properly and get everything applied on time. So we switched over to this, and unfortunately, we actually weren't able to apply that here at the Burlington Miscanthus site, just due to the volume of water kind of required to be able to dissolve these products into. But it was applied at the Guelph switchgrass site, which was really awesome to be able to have a good replacement for Listagrow. So getting into some of our results, I'm going to start with the effect of the treatments on plants. So I've summarized here just the yield data from each year at both the Burlington Miscanthus and Guelph switchgrass sites. We did also collect data on plant physical traits over the course of the season, but we didn't see too many really consistent trends or have like strong significance for the effect of the treatment, so I don't really want to spend too much time talking about that. Instead, we'll focus on the yield, and we didn't see actually any significant effects of the treatment in 2019, which is part of why we kind of switched up our treatments a little bit. But we did start to get a significant response in 2020, which I will focus on now. So here you can see at the Guelph switchgrass site in 2020, chemical nitrogen fertilizers, the urea, actually significantly increased the plant yields compared to the biofertilizers. So that's the combined average of all three biofertilizers, Jumpstart, Agtiv, and the Optimike and More treatment. So that kind of shows that maybe at this site, the chemical nitrogen is going to be the better bet in terms of increasing your yields. But that also contradicts some of the results that we got from Sean's thesis, his original trials, which showed that Jumpstart had higher yield than chemical nitrogen or controlled plots in his little smaller version of this trial. So I'm kind of thinking it might be a better idea to do some more research to figure out like under which conditions and what kind of years the biofertilizers might work better and which ones it would be better to use the chemical nitrogen fertilizers. But then moving into our Burlington miscanthus data, you can see that AgTiv actually almost doubles the yield of miscanthus compared to Jumpstart, the other biofertilizer. And it also, although not statistically significant, increased the yield by about 5 tons per hectare compared to the control and the chemical nitrogen plots. So that's a really promising kind of first look into the use of this product with miscanthus in a field trial. So skipping over some of the soils data just for a second, I want to touch on the climate data that we collected. So we had installed a couple of different chambers, at the Guelph switchgrass field site to measure the flux or the balance between the release and then consumption of carbon dioxide, methane, and nitrous oxide, which are all greenhouse gases. And I summarized kind of the average across treatments for July to August and September to October, so our summer months and fall months, for that data. And we did only get out there 1 day per month, just due to time and resource restrictions and reduction of research capacity. So you can see here that most of the emissions are coming from carbon dioxide, but that's not actually a horrible thing because that carbon dioxide is coming from soil microbial respiration. And soil microbial communities are really important to maintaining soil structure and supporting plant growth, nutrient cycling, decomposition. So that happens in all ecosystems on land. And it's also not taking into consideration the fact that the plants are kind of taking in carbon from the atmosphere as well. So this is just looking at respiration from the soil and not about all of that carbon that your plants are also taking in, storing in their roots in the soils and in their just above ground biomass as well. What you can see here too is the methane is actually negative. So that means that the soils are taking in or absorbing more methane than it's releasing during our sampling days. And it's not a huge number, but it is still taking that in, and it's important because methane is actually 21 times stronger as a greenhouse gas than carbon dioxide, so it's really great to be able to pull that out of the atmosphere. And then looking at the nitrous oxide, they have slightly positive, so it's releasing a little bit of that greenhouse gas, which is then 310 times stronger than carbon dioxide as a greenhouse gas. But those numbers are really, really small. So that's all really promising information to just look at how biomass crops are kind of affecting greenhouse gas emissions from the soil. And then the graph on the far side, I guess it's left for you, I think. I've summarized kind of all of the emissions data as a global warming potential. So if you take the flux rate for carbon dioxide plus the flux rate for methane multiplied by 21 and then plus 310 times the flux rate for nitrous oxide, then you can kind of convert it all into what it would essentially be if it was all carbon dioxide. So I've summarized that on the side there, and you can see for each of the treatments that the GWP, or global warming potential, is represented in the colored bars, and then the overall average global warming potential for each of our sampling days is in that black line. And we didn't actually find any significant differences here between any of the treatments on any of the days. But I think that if there was another research project that was done that had more time and resources to do more frequent sampling, you'd get more of the day-to-day variation that could capture kind of a more accurate and more detailed look at the seasonal variations and seasonal averages. And you might start to see some more significant differences there. But yeah, just off of our data, there wasn't too much in terms of significant differences. Moving now into some soil biology. For those of you that don't know, I think I already briefly mentioned soil microbes, and then also other kinds of soil critters and creatures are super important for supporting plant growth because they drive a lot of nutrient cycling, they support good soil structure, and they do a lot of other really important processes that support plant growth on all ecosystems on land on Earth. So for that reason, and because a bunch of our biofertilizers also added different microbial communities to the soil, we use genetic techniques to quantify the amount of bacteria and fungi that are present in the soils and how that's affected by treatment. So we did that at the peak season and at in the fall, near when we did the harvesting for biomass yield. in both study years, and we started to see significant effects more so in 2020. So in this first graph, you can see that both Agtiv and More significantly increased the abundance of bacteria in the top 10 centimeters of soil at the Guelph switchgrass site, and this is in 2020. And if I click over to our second graph, it's Agtiv and More again that have enhanced the fungal abundance at the same site, so at the Guelph switchgrass. And then these are both from the peak of the summer. So yeah, peak of summer, we've got higher fungal communities and higher bacterial communities in Agtiv and Optimic and More plots compared to our other 3 treatments. But those significant differences did disappear by the fall. However, microbial communities are a lot less active in the fall as well, so they kind of decrease in their activity due to changes in temperature. And we didn't actually see any significant differences or significant effects of treatment on bacterial or fungal communities at the Burlington Miscanthus site in 2020. But that doesn't mean that there weren't any other effects of these treatments on the microbial communities. They could just be effects that we couldn't capture by just counting the number of bacteria and the fungi that are present. So it could be changing the community structure, balance between different groups of bacteria and fungi, the diversity, the activity of different types of bacteria and fungi. So that could be another area for future research that would be worth pursuing, because those aspects are also really important in determining what the microbial community is actually doing and not just the size of it. But yeah, again, those data take a lot of time and effort and money to be able to collect, so we didn't have time to do that within the scope of this project. Moving into soil fertility, almost done, kind of. We had collected field samples at all of our sites in both study years to look at the soil and plant uptake of key plant nutrients. So it was the soil fertility in the top 30 centimeters, and then yeah, the plant uptake. So we collected plant tissue samples at the peak of the growing season to look at how the plants were actually uptaking those nutrients. And the main clear trend that we did notice was at the Guelph switchgrass site, plants that received the chemical nitrogen fertilizer had a higher tissue density, or tissue storage of nitrogen, which is not super shocking, because when there's more nitrogen available, they can uptake more and store more. But it was harder to observe kind of consistent trends and strong significant differences from these field data, just because there's going to be a lot of influence from the surrounding environment. There's the active plant growth to consider. There's interaction with the soils outside of our sampling depth, so beyond the 30 centimeters or outside of our plots. So that kind of makes things a little bit messier and harder to see. So to be able to control for that kind of expected messiness that comes with a field trial, we collected soils from the Guelph switchgrass field site for each of the treatments at the end of 2019. And we took the soils and put them into controlled conditions just by themselves with no plants or anything to look at the way that the treatments are affecting the soil availability of key plant nutrients. And that's where we saw that for our 4 fertilizer treatments from that year, soil nitrogen, phosphorus, and potassium were all almost universally increased compared to the control. So in this first graph, you're just looking at how each of those 4 treatments, the chemical nitrogen, Jumpstart, MicPro, and Listagrow, have higher soil mineral nitrogen compared to the control. Clicking over to the next one, you can see the same trend here for phosphorus, and this is from that incubation study, the controlled conditions study. Again, all 4 fertilizers have higher levels of phosphorus available. And then the third graph is for the mineral potassium, where again, 3 out of 4 of the fertilizers has a higher potassium level, with just Jumpstart being slightly lower. Not entirely sure why that is. But yeah, so it's really promising, and it shows that for regardless of which of these fertilizers you might use, they are improving soil fertility, which is of course the goal of doing fertilization. So I'm going to wrap up the presentation now just looking at some of the product pricing. So I'm not an economist, and a full economic assessment of these products would be outside of my expertise and outside the scope of this project, which was already really big to be doing in 2 years. But I've kind of summarized the cost of each of the treatments. in terms of how we applied them in 2020, except for Listagrow, which we only used in 2019, of course, and then the yield that they produced for switchgrass and miscanthus over the 2 years. So focusing first, on switchgrass, chemical nitrogen and Listagrow seem to be the most effective in terms of enhancing yields for this crop, and this is using only data from the Guelph switchgrass site because of some issues at the Burlington switchgrass site that kind of lost our yield data at the end of that season. But yeah, so chemical nitrogen is looking like it's kind of winning out here in terms of being the least costly option and also producing the highest yield. But again, we always have to consider then that overuse of chemical fertilizers can create a lot of environmental issues like soil and water pollution. It can disrupt your soil biological communities and result in higher greenhouse gas offloading into the atmosphere, especially right after you've applied it. So then you can look at Listagrow, which produced the second highest yield, and it is admittedly one of the more expensive options, but it adds a lot of soil organic matter and slower releasing nutrients that could be especially beneficial on lower class lands like classes 3 to 4, where these crops are being grown for the most part, because those class 3-4 lands could be experiencing limitations due to a depletion of the organic matter and certain nutrients. So it could be really beneficial there, and I think there needs to be more kind of look at how that can help to rehabilitate lower class soils. And now looking at miscanthus, so active, as it kind of came out in the yield data that I showed you, seems to be the most effective here, and it's only a little bit more expensive than applying just your traditional chemical nitrogen as urea. So that's really promising, and I think there should be a lot more research kind of looking into whether this kind of response with active being really successful in enhancing yield is consistent among different field sites, different conditions, year to year, and just some of the other benefits that it can instill onto the land. And then the second best, of course, and maybe not super surprising, would be chemical nitrogen. We've been doing it for years because it works. It did enhance the yield, so it has the second highest yield out of all the treatments and is also the cheapest. But then again, you do have to consider also some of the environmental impacts, especially if you're going for kind of a green economy and a renewable energy sort of agricultural system. So overall, thank you for listening to all of that. I hope I didn't go too fast or skip over anything. I want to say a huge thank you to all the people that have supported this work, including my advisors, all our funders, our landowners that allowed us to do our research here. Thank you guys both very much. And then all the companies as well who helped support us in guiding how to use their products and providing some of that to us. And I think the study is a really good first kind of look with a well-rounded view of how these different biofertilizers could be used for biomass crops. Yeah. If you have any questions, I can try my best.
Microbial products, if they were established in the soil, would you have to apply them every year, or do you think they could establish and then they carry on?
Yeah, that's something we weren't able to collect the data on that because it does take a lot of time and resources to like specifically identify which microbes are there, but it would be really great to do a follow-up study to see how long in the soil they're lasting and if they're able to reestablish themselves.
Very good question, Doug.
That was a great question, yeah.
Interested to go back without application this year and to collect soil samples to see whether we maintain the same microbial count. And what we are seeing is statistically there's noise in the data, but numerical values suggest. So we need to thank Professor Carrie Dunfield, who's in the center. She's the microbial prof. So what she's saying is sometimes the microbial community takes time to grow and establish itself in the soil profile. So she said it would be nice to see stop application in 2020 and then go 21, 22 to see whether these microbial populations still exist, or do we have to inject them every year? And if they do exist, then it's a great winning point for the landowners. They don't have to apply an inorganic fertilizer on an annual basis. And also the soil health. Now Cam is here, who is the soil health coordinator for University of Guelph, and that is what we are focusing on. See, the greenhouse gas emissions, why we measured was that we have to take into account the carbon capture as well. So the long term, we're trying to establish that the biofertilizers will reduce greenhouse gas emissions, the phosphorus, nitrogen loading to the waterways, and still maintain biomass yield. So from Sarah's work, I mean, it shows the active and Lister-Grow. I mean, these are the winning, and then also Optimic. You know, we are having some winners here, but we need some long-term research to establish the fact that they can colonize, they can exist, they can overwinter. These are the questions that we still need to answer. But then, unfortunately, we can go back because the province took the land. It's for sale now. Yeah, it's for developing, for development. So that's one of the challenges of research, right? Long-term research. But we can count on Norm. You know, he always allows us to. He's not going to sell the land and kick us out. Any more discussion? You can. We can transfer that.
Is there any vision, like with the very last slide I showed with miscanthus, where the yield actually exceeded the nitrogen? The nitrogen rate is a little bit low, I guess. At a full nitrogen rate, would you expect there to still be a yield bump? Or is there a vision that this stuff could actually replace a higher nitrogen application? Like, how do you sort of understand the opportunity there to get away from commercial nitrogen?
Yeah, so that nitrogen rate there is actually the recommended rate for these products. They don't, or for these crops, they don't really require a whole lot to even begin with. So 60 kilograms of nitrogen per hectare is a pretty standard rate. With AgTIP, the way that it works is it just enhances the uptake of nutrients that are already there. So there might be every so often you need to like add an organic fertilizer like a manure or maybe something like Lister-Grow to be able to supplement, because you're just going to be taking more nutrients that from the soil without necessarily adding with those microbial treatments. But yeah, I'm not really fully sure how to answer your question. I guess you could. Go ahead. Yeah.
Good answer. So we really don't have solid data on miscanthus for an optimum nitrogen rate. It is a wrecked crop. Unlike switchgrass, we know if we go too high, the crop can lodge. With miscanthus, especially because there's several distinct varieties with different yields, we don't have that. What is the optimum nitrogen rate? How much can we put on to get the yield increase in Ontario conditions?
Yeah.
So it's a bit nebulous, but certainly, we can see here the, increased yield from the product. Would we get that increase in nitrogen as well, going over the recommended rate? That would take more research. And it would be like the genome varieties are much bigger than the switchgrass. So we have, the gigantus is different than the stuff we have at Fort Ryerson. David Smith is propagating on the Ryerson variety. And then we have the one that Dean Thiessen brought in, again, which is a different variety, so we don't have a clear answer to that, but certainly what's promising what's up there.
Yeah, and maybe Norm, we can try the active and see miscanthus plots whether it's giving any response, and then not apply nitrogen fertilizer for 1 or 2 years and see the response. You're welcome to try. And please help yourself. There's still coffee. Just be free to go and help yourself. Any more questions?
Yeah. Hi. Thank you.
Just on the biofertilizers, are they fungal based or bacterial based?
So most of these ones are fungal. So the Jumpstart and AgTIP are fungal, and then Optimic is a mixture of different funguses, or fungi. I guess that's how you say. And then more is a bacterial base. So that's a mixture of different bacteria, but it just depends on the product.
So when they are mostly fungal based, then how are you comparing with the nitrogen? Because they are mostly for phosphorus solubilization.
That was Jumpstart is more for the phosphorus solubilization. But AgTIP, it's an arbuscular mycorrhizal fungus, so it'll help them to access any nutrients that are in the soil. So the Jumpstart product, Penicillium villi, is a phosphorus solubilizing bacteria, fungus, sorry. So that's the one that'll focus more on phosphorus uptake, but AgTIP does help with nutrient uptake across the board. if
And
that
this is,
answers.
yeah, sorry, and this is where the natural climate solution funding should come to the growers. So you can see the cost, and if any grower is contributing to natural climate solution management practices, that difference in cost should be covered through the natural climate solution funding. So this is where we need federal government and provincial government incentives to promote landowners to go green and still achieve, and then substitute this is what is happening in Europe. They are getting, even for biomass, for energy purposes, European governments are really giving the incentives for the landowners. And so this natural climate solution, it is currently is like living lab, entity, right? But it should come out of that, and then any biomass grower, any crop grower who is depending on natural fertilizers, biofertilizers, contributing to pro-environmental practices, should register with the natural climate solutions and then derive the cost difference and as an incentive. So this is something that we should promote the governments to